I wanted to welcome all of you here in the room as well as on the Zoom. Hopefully you're not saturated by presentation at Arvo, and I see that most of you were able to come back on time to attend this seminar in person. So that's wonderful. And today we have a really important topic that wasn't that well known just a decade ago, but now is a major hub for mutation and diseases and so on. And we have a really expert who work in this area for a while. So with that, let me ask David to introduce our speaker today.
It's my pleasure to introduce our distinguished speaker for today, Dr. Gregory Pazour. Dr. Pazour is currently a professor of molecular medicine at the University of Massachusetts Chan Medical School. The Pazour group is interested in the function of the intraflagellar transport proteins and the role that cilia play in development and in degenerative diseases like blindness and polycystic kidney disease. He earned his PhD from the University of Minnesota, where he studied the plant pathogen Agrobacterium. This was followed by a postdoc at the Worcester Foundation for Biomedical Research, where he made the unexpected connection between cilia in the green alga Chlamydomonas and polycystic kidney disease, PKD. In 2002, he showed that the gene PKD2 localized to cilia, explaining why cilia were critical to prevent cyst formation. Since then, cilia dysfunction has been identified in other genetic diseases, including blindness, congenital heart disease, skeletal abnormalities, and cognitive impairment. Collectively, these diseases are known as ciliopathies. The contribution of Dr. Pazour has been recognized with several awards, such as the Kaplan Prize for the International Society for Nephrology, as well as being named in 2019 a Fellow of the American Society for Cell Biology. Please join me in welcoming Dr. Greg Pazour, who will be presenting his talk, Cilia, Little Organelles with Big Roles in Development and Disease.
Thank you so much for that great introduction, and also for inviting me here. It's a real pleasure to have this opportunity to come and talk to you guys about the work that my lab has been doing to understand the assembly mechanisms of eukaryotic cilia and the role that these organelles play in diseases like retinal degeneration, polycystic kidney disease, and other things. So what I'm going to do today is I'm going to start out with a fairly extensive background on cilia, a bit of history of how I got involved in this. We'll have a little bit of retina in there, but a lot of basic cell biology of cilia. Then we'll move on and we'll talk about some work that we've been doing more recently to look at the intersection between ciliary assembly mechanisms and hedgehog signaling during development. So the organelle, how do we get this to advance? There we go. The organelle that we're going to be talking about today is called cilia or flagella. These are these whip-like or hair-like organelles that stick out from the surface of many types of eukaryotic cells. These are microtubule-based organelles. The microtubules are templated from the centriole. You may be more familiar with the centriole being part of the spindle pole during mitosis, but when the cell is not undergoing mitosis, it extends microtubules off the end of that centriole, and those microtubules form the cytoskeleton of the cilium. They extend along the entire length of the cilium. So if we take a cross-section through one of these cilia, we get this classic arrangement of microtubules called the 9+2 arrangement. What I'm going to tell you today is that cilia do two big things in eukaryotes. First of all, they produce force to allow single-celled organisms like this green algae here to swim through liquid medium. In multicellular organisms, they often move fluid over the surface of the cells. And then they also serve as sensory organelles that allow cells to sense the environment. So many of those proteins that are needed for those two functions are anchored onto the cytoskeleton. So for example, this structure right here is an outer dining arm. It's permanently attached to this microtubule. It reaches across and makes a transient interaction with this microtubule over here and causes these microtubules to slide with respect to each other. And that's how the force is generated to move the cilia. So the motility functions are often attached to the microtubules, but also sensory functions are bound onto here as well. Sensory proteins are bound here as well. And then this cytoskeleton is surrounded by an extension of the plasma membrane that you can see here in this EM called the ciliary membrane. This is an interesting domain of the plasma membrane. It's continuous with the plasma membrane of the cell, but the cell can put receptors on the ciliary membrane that it doesn't put on the cell body. And that, of course, is incredibly important in the sensory functions of cilia. So cilia and flagella are very ancient organelles. They were present on the last common ancestor of all currently living eukaryotes. And then over that about 2 billion years of time, these things have become evolved to do all sorts of different motility and sensory functions. So for example, Chlamydomonas swims around with two motile cilia at the end of the cell. It sort of does the breaststroke to pull itself along. It also uses these as sensory organelles. And the best studied example that we know of is during mating. There's a plus and a minus mating type. The two cell types come together, their cilia adhere to each other, and then a signal is propagated from the cilia down to the cell body, which causes the cell wall to come off and then the cells to fuse so they can undergo meiosis. When you get to other organisms like C. elegans, the nematode, they got rid of all of their motile cilia, but they kept non-motile sensory cilia on their dendrites that they use for sensing the environment during mating and during feeding and other behaviors. And then when you get to more complex organisms like vertebrates and humans, you can find all sorts of different cilia within the same organism. And so for discussion purposes, I like to break these into these two large categories of the motile and the primary/sensory cilia. But keep in mind that I just told you in Chlamydomonas, the motile cilia are sensory, and that's also true in mammals as well. But these categories are somewhat useful in thinking about the diseases that result from defects in cilia. So if we start first with the motile cilia, these are very abundant in your respiratory tract. You can see a clump of them here on this tracheal epithelial cell. And these things beat and they push a mucus layer over the surface of this epithelium. That mucus layer will trap bacteria and particles that get down into your lungs and move them out. And so patients who would have defects in this motility will end up with a degenerative lung disease called primary ciliary dyskinesia. These are also very prevalent in your brain. They line the ventricles and the canals that connect the ventricles together. They seem to be important in moving cerebral spinal fluid over the surface of the ependymal cells. This has some connection to epilepsy. We don't really understand the mechanism, but a number of motile cilia genes also are epilepsy susceptibility genes. And they also are very important in preventing hydrocephaly, which is the accumulation of cerebral spinal fluid in the ventricles and the enlargement of the brain. So in dogs and rodents that have motile cilia defect, almost 100% of them will get hydrocephaly. In humans, it's a risk factor, but not nearly as prevalent as it is in dogs and rodents. These are also very prevalent in the female reproductive tract, where they're thought to help move the egg along. And then, of course, the male gamete, the sperm, swims through the female reproductive tract by the action of the sperm flagellum, which is a motile cilium. So the other part of this category would be the primary/sensory cilia. When I started to give this talk back in the last century, we didn't really know what to do with primary cilia. But we knew at that point in time that there were two cilia in vertebrates that clearly served as sensory organelles. One of them you're very familiar with in this audience, the other being in your nose, where the olfactory receptors are concentrated in olfactory rods or olfactory cilia. And the first steps in olfaction occur within those cilia. Of course, as you're well aware here, in photoreceptor cells in the retina, the outer segment, which would be the light detecting part you see here with these opsin-containing bands, is a modified cilium. So this part up here, the outer segment, is connected to the bottom part, the inner segment, by this little bridge here, which is the connecting cilium. This is the part of the cell that has the biosynthetic material in it. So this whole part here is all a very highly modified cilia. And the reason that I say that is to build this structure, you start with a cilium that looks like one of these over here. The magnifications are different here, but this diameter right here is the same as this diameter here. Then you move membrane and proteins into that cilium. These discs form from the ciliary membrane and push out. So this entire outer segment is a very highly modified cilia. The fact that you can see this slide is due to the sensory perception within your cilia in your retina. So the other part of the category here, the primary cilia, we now know are sensory organelles, and we'll talk more about that as we go along. These are distinguished from the motile cilia by the fact that there's only a single one per cell. There's typically non-motile as well, although there's one example that we know of where we've got these solitary cilia that are motile. That occurs in vertebrate development on the node. So the early vertebrate embryo is left-right symmetric, and then during development, a little pit forms that's covered with monocilia that have sort of a rotary motion to them, and they push something to the left side of the embryo. What's actually getting pushed is controversial in the field, but The result of that is that you get differences in gene expression on the left and the right side of the embryo. That gives rise to our left-right asymmetric organization of our internal organs. So our stomach is more on the left, our spleen is on the left, but it really plays a critical role in heart development. So we need to separate our left and right sides of our heart so we can oxygenate blood in the lungs and then ship it back out to the body and bring deoxygenated blood back. So somewhere on the order of about 1% of babies are born with some structural heart defect, and a lot of those are being caused by defects in this motility in a disease that's called heterotaxy. There's also another condition called situs inversus, where you can be completely reversed in your internal organs. That's a pretty benign condition. It's estimated that something like 1 in 2,000 people are walking around with their internal organs reversed and not even know about it. So we'll talk more about the primary cilia, but essentially they're found very widely throughout the eukaryotic and throughout vertebrates and are thought to be sensory organelles. So the question that my lab has been interested in now for quite some time is, how do you build one of these structures? And the problem is exactly the same whether you're thinking about a flagellum on a chlamydomonas cell, primary cilium on a cultured cell, or even something as complex as a rod or cone outer segment. And that is, you've got a very complex organelle. There's somewhere on the order of 1,000 to 1,500 unique proteins in cilia. Some of them, like the tubulins and the dyneins, are very abundant in cilia, but there's no protein synthesis capacity here. All of the proteins are made in the cell body, and then they have to be transported into the cilia to build and maintain these structures. So there's a few things that conspire against this being a diffusion-driven process. One is the distance. So proteins are made here, but the cytoskeleton is assembled at the distal tip. So in chlamydomonas, that could be 10 microns away. Other cases, it might be 50 microns away between the sites of assembly and the sites of synthesis. In addition, there's a time problem that chlamydomonas flagella assemble quite quickly. If we knock these off, they can grow back in about an hour's time. So these things coming together suggested that we needed some sort of an ATP-dependent, motor-driven process to move the materials made here into the cilia to build and maintain these structures. So this is the process that we think drives ciliary assembly. It's the process of interflagellar transport. It was first described in chlamydomonas by Joel Rosenbaum's lab. What you're looking at here is one of the two cilia from a chlamydomonas cell stuck down on glass, with the cell body being up here out of the field of view. You should be able to see particles being trafficked from the cell body to the tip and other particles being returned back from the tip back to the cell body. This movie is running about real time. The particles move out at about 2 microns per second, carried by kinesin 2. They're carried back at about 4 microns per second, carried by dynein 2. So these particles we don't think of as structural components of cilia, but we think of them as motor adapters that allow the single pair of motors, one kinesin, one dynein, to carry 1,000 or more cargos from the cell body into the cilia to build and maintain these structures. So something that needs to carry 1,000 or more different cargos is probably going to be pretty complex, and it is. This is a current view of what we think is moving in the movies I just showed you. So we think those particles that are being trafficked along are large complexes made up of 3 subcomplexes: IFT complex A, IFT complex B, and the BB-some. So A has 6 proteins in it, B has 16, the BB-some has 8. And then there's some additional adapter proteins. So for example, TULP3 is thought to work with complex A to move membrane proteins into cilia. OTA16 works with complex B to move outer dynein arms into cilia. There's also another part of the complex where one of the IFTs is found at the Golgi in association with the Golgin. We think this probably has something to do with trafficking between the Golgi and the cilium, but has not been fully established. So my involvement in the IFT system came in the late 90s when I identified a mutation in this gene right here, IFT88, that's part of IFT complex B. So when I joined George Whitman's lab at the Worcester Foundation to do a postdoc, what I did is I carried out a large-scale screen in chlamydomonas looking for cell lines that had either defects in ciliary assembly or ciliary function. And I identified this particular line that's shown over here that's completely missing cilia. The cilia should be coming out of these collars right here like it is in the control animals. These guys completely lacked cilia. So we identified the gene that was defective in here, and it turned out to be the IFT88 subunit of IFT complex B. That in its own right was a really important finding because Doug Cole, working with Joel Rosenbaum, was purifying the IFT particle. And this was the first evidence that these proteins that were moving along the cilia actually were required for ciliary assembly. But the really exciting thing came when we took the sequence of it and blasted it against the database and realized that there was a mouse gene out there that was known called the TG737 gene, or Oak Ridge polycystic kidney disease gene. So this gene had been identified in Rick Wojciech's lab at Oak Ridge National Laboratory in an insertional mutagenesis screen in the mouse. And they found that the mice that were mutant for this particular gene failed to thrive, and they usually died shortly after birth, but the ones that survived into adulthood developed polycystic kidney disease, as well as cysts in their liver and their pancreas. So they cloned the gene that was defective, and it didn't look like anything at that point in time because our chlamydomonas work had not yet been done. So there wasn't a lot of progress made in understanding the cell biology of the TG737 gene, but it did become one of the models for cystic kidney disease. This is a really important human disease. It's the most prevalent single gene disorder of humans, affecting somewhere on the order of 1 in 1,000 people worldwide. It's typically described in the textbooks as being caused by the excess proliferation and poor differentiation of the kidney nephron epithelium. So of course, blood is filtered in the glomerulus, and that filtrate moves through the tubules of the nephron and the collecting duct. All of these tubules are lined with a single layer of epithelium. And in cystic disease, that epithelium overproliferates, and it turns these tiny tubules into these large fluid-filled cysts that you can see over here that continue to enlarge and destroy the kidney. So a normal human kidney is about the size of your fist. These kidneys will be the size of a football or larger and can impede people's ability to breathe. They become so large. So we knew that IFT88 was highly similar to TG737, likely an ortholog based on sequence conservation. We knew that TG737 was required to prevent this nasty disease that you see here, but there was no evidence in the literature that TG737 did anything with ciliary assembly. So we obtained these mice from Oak Ridge, and we took a look at their kidneys by scanning EM. What you can see here in the wild type collecting ducts is that each of these epithelial cells has a nice primary cilia sticking out from it in the controls. If you notice the mutants down here, you do see a few little remnants there, but for the most part, the cilia are missing. It turns out this is actually a hypomorphic allele. As I'll tell you later, if you're completely missing cilia, the animals would have died mid-gestation well before the formation of a kidney, and we would have never been able to see this disease develop here. So it's a very special allele there. So I remember the day that we took these pictures. I knew at that point in time that we were going to change the view of the primary cilium, because this image clearly showed that defects in primary cilia had something to do with polycystic kidney disease. And up to that point in time, the cell biologists either didn't know about the primary cilium, or if they did know about it, they had dismissed it as being unimportant and uninteresting and not something to waste your time on. But this said it did something important in maintaining tubule diameter. At that point in time, I had no idea how big the primary cilium was going to become, but I felt we were going to change the view there. So at the same time, when we realized that the TG737 had ciliary assembly defects, we knew, of course, that rod and cone outer segments were modified cilia, were developed from cilia. And so we reasoned that perhaps the same process that builds a primary cilium in the kidney could be involved in building a rod or cone outer segment. So I worked with Joe Bashars at the time, and we took a look at how this mutation affected the formation of rod and cone outer segments, or I should say we focus on rod outer segments. And what we found in the mutant animals is that early after birth, they really failed to form outer segments in any significant way as compared to the controls over here. And the outer segments that did form degenerated fairly quickly, and then we lost the retinal layer as well. So the gene was clearly important in forming the rod outer segments, and its loss led to retinal degeneration. So I think of those two diseases, cystic kidney disease and retinal degeneration, to be two of the founding members of what we now call the ciliopathies. This is a huge collection of diseases. I've got listed some of them over here on the left. I'm not going to go through the whole list, but what I want you to appreciate is that defects in cilia lead to either developmental or degenerative diseases in pretty much every organ that has been examined. And so patients can have either isolated disease in a single organ, or they can have syndromes where multiple organs are affected. And it's been estimated that the human literature has at least 200 syndromes described that probably have ciliary dysfunction as the primary cause. So whether a mutation causes an isolated disease in a single organ or causes a syndrome is thought to be related to two things. One being the function of the gene, and the second being the strength of the allele. So with regard to the function of the gene, what we think is that if the gene is only needed in the kidney, we're going to end up with cystic kidney disease. If it's a retina-specific gene product, we're going to end up with blindness or retinal degeneration. If it's needed both in the kidney and the retina, we're going to end up with something like senior-Loken syndrome, where you have retinal degeneration coupled with a type of cystic disease. And then if it's needed more broadly, you'll end up with these other syndromes here. And then in addition to that, the strength of the allele seems to be really critically important in Cilia genes. We've got a number of examples now where different alleles will give us different diseases. So one of my favorites is IFT140, which is part of the IFTA complex. Isolated mutations in that gene will give you male infertility. Other ones will give you cystic kidney disease. Others will give retinal degeneration. And then still others will give syndromes over here in the Bardet-Biedl and skeletal dysplasia sorts of syndromes from there. That's thought to just be related to the particular mutations in these different alleles and may reflect the cargoes that are being transported by that particular protein. So my lab's been interested in trying to understand the function of the IFT proteins. And sort of the intellectual drive behind this is the high degree of conservation of these proteins. So the IFT system is conserved in every eukaryote that builds a cilium in a membrane projection from the surface of the cell. And these proteins here were initially identified in Chlamydomonas, but every one of them has a clear ortholog in humans. And in many cases, it's about the same size in humans as it is in Chlamydomonas. That's pretty remarkable because Chlamydomonas and humans shared a common ancestor about 1.6 billion years ago. So that tells me that each of these proteins has some important function to play in ciliary assembly or ciliary function. And when you look at the sequence of these genes, there's a lot of different motifs and different activities encoded within here, suggesting that there's a lot of individual functions to sort out. And so we've been trying to do this by making knockout mice and then looking at the phenotypes of those animals to try to deduce what the functions of the particular proteins are. And I'm going to spend most of my time today talking about these two proteins here, IFT27 and IFT25, but they're rather atypical IFT proteins. So before I get to those, I want to tell you a bit about IFT20, which is a more standard IFT gene, to sort of give you a flavor of what happens when you knock out most of the IFT genes. So as I told you already, that the TG737 hypomorph was survived through development, but if you made a null allele of that gene or any of the other genes that are completely required for ciliary assembly, you end up with a mid-gestational lethal phenotype. And so the alleles that we've studied over the years have typically been floxed alleles. And so here's our IFT20 floxed allele, where we've got a LOXP site in intron 1, another one in intron 3. These mice are completely normal. Then if we cross these mice to a Cre that's expressed in the germline, we'll delete these 2 exons. It takes out the only methionine, so very highly likely to be a complete null. This gives us an embryonic lethal phenotype at about day 10.5. You can see examples of this down here on the bottom. So on the left, we've got a 10.5-day-old embryo here. It's undergone embryonic turning. The heart is looped and it's beating, moving blood throughout the embryo. The mutant, on the other hand, has failed to undergo embryonic turning, and it stays in this lordotic or backwards S configuration. You'll notice the heart here. It's failed to loop, and it's projected straight out from the heart, or excuse me, straight out from the chest, and it's surrounded by this pericardial effusion here. So cilia don't seem to be real critical in early development, but they are needed for organ development. So when the embryo gets large enough that it needs a heart to move blood around and distribute nutrients and oxygen, that's when the embryos fail from there. So the beauty, of course, of a floxed allele is you can delete in different cell types and get around this embryonic lethality. I want to show you 2 quick examples here of where we've done this with IFT20. So first we did it in the kidney using a Cre driver that's expressed only in the collecting ducts, which are marked in the brighter green here. So in the control animals, you see the collecting ducts here. These are 2 layers of cells with a tiny lumen in between them that you can barely see at this magnification. This is the normal condition, but if we take out IFT20, you see that these lumens have become massively enlarged due to the cystic expansion of these cells here. So in this case, if we zoomed in, each of these cells would be ciliated. In this case, the cells are not ciliated there. So very typical of the loss of cilia in the kidney. We also used the human red-green opsin promoter to delete the gene in photoreceptor cone cells. And you can see the staining with the red-green opsin here on the left, nicely in the outer segments. in controls, whereas when we knocked out IFT20, the first phenotype that we saw, of course, was the redistribution of that opsin into the inner segment and down around the nuclear layer. And then with time, that led to degeneration of the cones and loss of the cones. So that's a phenotype that's been seen by lots of subunits of IFT complex B. IFTA is similar, except that this is not quite as important for ciliary assembly, and so these animals tend to live a little bit longer, and the disease phenotypes are a bit less extreme there. So what those phenotypes say to me is that those tell me that these proteins are needed for ciliary assembly, and those are cilia phenotypes, not necessarily giving us any individual function of the subunits in here. And that's really what we're interested in. So in the last years, we've been focusing more on cell culture, where we can make more subtle mutations rather than just a null allele to try to tease out some functions there. I'm not going to tell you about that, but instead I'm going to tell you about this pair of proteins here. And the reason that I focused on these, before I start that, IFT27 is a small G protein distantly related to the Rab family of proteins. IFT25 is a calcium binding protein. These two proteins form a stable heterodimer that's part of complex B, where IFT25 wraps around 27. So the GTP binding and the calcium binding are interesting regulatory or potential regulatory mechanisms of IFT, so it makes them interesting in their own right. But what was really interesting to me about these guys was their evolutionary distribution. So I'd said that the IFT proteins are highly conserved across eukaryotes, and that's particularly true for IFTB, where you see them in Chlamydomonas, you see them in vertebrates, you see them in C. elegans, you see them in Drosophila. But if you look at enough organisms and enough IFTs, you'll find one or two missing here and there. And what I notice is that these two proteins that are part of B, form a stable heterodimer, are present in Chlamydomonas, present in vertebrates, but missing from C. elegans and missing from Drosophila. And so I reasoned that the fact that these organisms can build cilia without these two proteins would suggest that maybe these aren't needed for ciliary assembly, but maybe they're needed for some sort of sensory functions that aren't needed in C. elegans and Drosophila. So we obtained cells from the COMP project and made knockout mice for each of these. The alleles are shown up here on the top. In both cases, we're missing at least one exon, and we've got a gene trap insertion in there to capture the upstream exons if they're transcribed. We think we've got null alleles or very close to null alleles based on both RNA and protein work. The proteins are shown down here on the bottom, where we've got Western blots from controls, IFT25s, you see 25 and 27. 25 is gone, 27 is mostly gone. In the 27 knockout, 25 is still present, but 27 is missing from there. So the knockout of these two animals is very similar to each other. And so as I go on for the rest of the talk, I'm going to jump back and forth between 25 and 27 and just show you one image, whichever one I have a prettier picture for. So both of these animals have a lethal phenotype, but it's very different than the lethal phenotype that I just told you about with the IFT20 or many of the other IFTB genes, where they die mid-gestation at the time when the heart is starting to beat. These guys make it through development. They're born alive, and then they die within a few hours of birth. You can start to appreciate some phenotypes here. These guys are a bit smaller, both 25 and 27. Maybe you can appreciate some craniofacial abnormalities here. They have almost completely absent lower jaw. They have sort of a bluish cast to them, which is called cyanosis. So the fact that these animals made it through development alive, or made it through development and were born alive, suggested to us that they probably didn't have a severe ciliary assembly defect. Because every example I know of where cilia are completely gone, the embryos would have been dead long before that point. But we took a look at that in a couple of ways. One shown up here on the top, where we looked at cultured fibroblasts derived from either wild type or mutant embryos. You see the red cilia in the control, and in the mutant, you can see green IFT27 in the control, but not in the mutant. We look in populations, the length is about the same, the percent is the same there. So we don't see any defects in ciliary assembly in these fibroblasts. If we look in the embryo, you can see the cilia in all the normal places. Here's the airway in wild type and mutant. Here's a kidney tubule in control and mutants there. So the animals don't seem to have ciliary assembly defects, but remember that they die at birth, and they have a whole constellation of phenotypes. A few more are shown here on this slide. So up here, I'm showing you a cleft palate. So this bone right here would be the bone. If you stuck your finger in your mouth and touched the top of that, that bone is missing in the mutants, and then the side bones have grown in to fill in that space. There's also a lot of polydactyly digit defects. You can see an extra thumb here, an extra middle digit in this case. And then many of them have their higher number digits fused together. And that's associated with the clubbing phenotype where the paw is sort of bent there. They have a little bit of a left-right patterning defect, as you can see down here. So in the mouse, there's a single lobe on the left side with a multi-lobed right lung. These guys have a single lobe on both the left and the right side. And so I had showed you that cyanosis, that bluish cast to the embryos. That's typically caused by a failure of the blood to be oxygenated. It could be due to lung or heart defects. This is probably not sufficient to cause the cyanosis, but there is a pretty significant heart defect that's likely to be responsible for that cyanosis and probably the postnatal death. And that is that they fail to separate their left and right atria from each other. So normally there'd be a septa in the embryos to separate the left and the right atria from each other. These guys, there is no septa. So the blood that's been oxygenated coming back from the lungs mixes with the deoxygenated blood coming from the body. And so this is going to be a very inefficient at moving oxygenated blood out to the embryo. And it's probably the reason that the embryos are blue at birth. So when you look at this collection of phenotypes, what immediately stands out is this is a collection of phenotypes that you see when you have weakened hedgehog signaling. So many of you are familiar with hedgehog signaling. It's one of the classic pathways that was identified by Nussbaum, Vohard, and Wieschaus in their Drosophila screen. This pathway is also important in vertebrates. So strong alleles in vertebrates will lead to mid-gestational lethality, whereas weaker alleles will lead to things like are shown here in this particular mouse. I should point out, so finding the hedgehog phenotypes in our mouse was really exciting to us because in the previous years, work from a number of labs started by Catherine Anderson, Brad Yoder, Jeremy Ryder, and others had shown that in vertebrates, the hedgehog pathway is organized around the primary cilium. So this is not true in Drosophila, but it's true in vertebrates. This cartoon shows our current understanding of how the hedgehog pathway works in vertebrates. So at the basal state, so missing sonic hedgehog or any of the hedgehog ligands, there's a 12 transmembrane receptor called patch that sits in the ciliary membrane, produces a negative signal that inhibits a GPCR called smoothened and keeps it from coming into the cilia or accumulating in the cilia and keeps it inactive. When you add sonic hedgehog, that binds directly to patch leaves the cilium, that removes that inhibition on smoothened, causing smoothened to accumulate in the cilia. Then it becomes activated and activates the Gli transcription factors. So the Gli transcription factors are typically found at low levels at the tip of the cilia, the basal state. Then upon pathway activation, they become very highly enriched in the cilia. It's thought they're processed here before they then move down to the nucleus to control genes that are under the control of this particular pathway. So our mutants look like weakened hedgehog signaling, but we wanted to look at that more directly. And a good way to do that is just to measure the expression of the GLI1 gene. So this is a transcription factor in the pathway, but it's also the most upregulated gene in a amplification mechanism. So under normal conditions in a wild type cell, absence pathway activation, in this case, we used an agonist of the pathway. You have very low GLI1 expression. If you activate the pathway, you get high levels of GLI1. If you look at our IFT25s, the basal state looks about the same, and we do get some induction, but it's about 1/3 what you would get in the controls. You can see the same thing happens in the 27 mutants. So interestingly, this is about the same thing that happens if cilia are missing. So cilia aren't needed for all types of hedgehog signaling, but they're needed when you need high levels of signaling at certain points in development, like during craniofacial development, heart development, and digit formation. So our mice are behaving as though they're missing cilia with regard to hedgehog signaling. But remember, these guys have no defects in ciliary assembly as far as we can tell. So that caused us to think about a different model where what we thought was happening is that maybe IFT25 and 27 that are part of this IFT system that's running continuously up and down these cilia aren't needed for assembly of the cilia, but maybe they're needed for these dynamic movements of patched and smoothened that occur during signaling. So to test this idea, we looked to see how patched and smoothened were affected in our mutations. And we'll start first with patched. So patched is the 12 transmembrane repressor of the pathway that's normally found in cilia at the unstimulated state. So here's an unstimulated wild type cell. You see the red cilia there. You see the green patched in that cilium. If we then add sonic hedgehog to that, we can now see this patch leaves those cilia, and we can quantitatively see this reduction in signal there. If you look at the 25s or 27s, you see the cilia there, and you can see the patch there. We don't see any quantitative difference between these two populations. But then notice over here, when we activate the pathway, the patch remains in the cilia. So there doesn't seem to be any problem in getting patched into the cilia, but it's the regulated removal that is defective in our particular mice. So smoothened shows the opposite phenotype. It's normally missing from the cilia. When you activate the pathway, it becomes enriched in the cilia. If you look at our mutants, what you see, though, is that we've got smoothened in the cilia at the basal state. And it remains there and actually goes up after pathway activation. So very similar to patch, there's no problem getting smoothened into the cilia, but it's the regulated removal that is defective there. So work from a number of people from my lab came up, and we built this particular model that's shown here, where what IFT 25 and 27 are doing is that they're coupling the IFTB complex, so the IFT particle, to the third part of the complex, the BB-some, for this removal of patched and smoothened from the cilia. So a lot of details went into figuring this out. I'm not going to go into them, but what we focused on at that point in time was, how does the BB-some working through the IFT complex know to remove smoothen from the cilia at the basal state, but let patch remain there, and then after pathway activation, allow smoothen to stay and now remove patched. And so we started to think about that, particularly in regards to smoothen, this GPCR, because the GPCR literature has a lot of examples of where receptors are found on the cell surface when they're in the inactive state, but then get internalized after activation in order to shut down the pathway. We thought smoothen being a GPCR, maybe it uses a similar mechanism to regulate its ciliary levels, even though the pathway is backwards and that it remains in the cilia after activation. So in the GPCR literature, there's two main mechanisms that regulate whether the receptor is on the surface or been internalized. One is a beta-arrestin dependent mechanism, where upon ligand binding, the receptor gets activated, that causes it to get phosphorylated, and then that phosphorylated residues bind to beta-arrestin and it gets internalized. An alternative pathway is through ubiquitination, where the activation of the pathway triggers ubiquitination and then internalization from there. So to test these ideas, we started first with the beta-arrestin pathway. And what we did is we knocked out the two forms of beta-arrestin that we would expect to be involved in regulating smoothened levels, and then measured the amount of smoothened positive cilia. So smoothened in a wild type cell is very low at the basal state and then becomes almost 100% of the cells are positive after pathway activation. And so we reasoned if this pathway was important at the basal state in a beta-arrestin knockout, we should see a lot of smoothened positive cilia. We do have a few here, but it's nothing like what we would have in an IFT 25 or IFT 27 mutant. So we don't think this is a very important part of the pathway regulating ciliary smoothened levels. So that caused us to focus on the ubiquitination pathway. The ubiquitination pathway, of course, is this multi-step process where a ubiquitin is picked up by an E1, transferred to an E2 conjugating enzyme, and then that E2 works with E3s to put those ubiquitins onto substrates. That gives you monoubiquitin. The monoubiquitinated substrates can then be further extended on different lysine residues within the ubiquitin to get you polyubiquitin. And the type of chains that are produced control the fate of the substrate. So if you build off of K48 lysines, you get proteasomal degradation. If you build off of K63, you'll end up with defects in signaling and complex formation, other things like that. So in the Drosophila literature, so Drosophila have robust hedgehog signaling, but they don't use cilia for it. In fact, the cells that do it aren't ciliated, but instead they move between the cell surface and internal stores. And it's known in Drosophila that at the basal state, smoothened is ubiquitinated at a higher level than it is after pathway activation. So we were interested in determining if that was also going on in the mammalian system. So to do that, we created two constructs. One is where we took the smoothened and we tagged it with a flag, and then we took ubiquitin and tagged it with an HA tag. We co-expressed those in cells, and then we did immunoprecipitations either at the basal state or after pathway activation by sonic hedgehog. You can see in both cases that we got about the same amount of smoothen pulled down, so there doesn't seem to be a change whether the pathway is on or off. Then we also made another construct I'll show you in a few moments, where we took all the intracellular lysines and changed them to arginines in order to completely prevent ubiquitination. You can see that stabilizes the protein here, indicating that at least some of the smoothen goes through the proteasomal degradation pathway. And again, there's no difference between the basal and induced state. So the question then is ubiquitin incorporated more at the basal state than at the induced state? And so to do that, we probed these same IPs with HA to detect the ubiquitin. You can see this chain of ubiquitin here at the basal state. Then you could probably appreciate that after pathway activation, there's a reduction, particularly in these higher molecular weights here. If we quantitate that in multiple experiments, we routinely saw a reduction in the amount of ubiquitin there, and essentially no ubiquitin incorporated in the no arginine or no lysine controls, saying this really is lysine ubiquitination from there. So that's consistent with a model where smoothen could be ubiquitinated at the basal state, and that made it cargo for removal. But we wanted to test that further. So we took another clue from the GPCR literature, and we put a ubiquitin on to the 3' end of the open reading frame so that we could see the fate of this ubiquitinated residues, or excuse me, ubiquitinated GPCR. So we have this pair of constructs, one being the smoothened with a flag on it, and then the other one with the flag and the UB. If we take these then and put them into cells, the wild type cells expressing smooth flag, you see the green cilia, the basal state, there's no protein there. After pathway activation, it's enriched there. You can see the quantification over there. If we put that into 27 mutants, we see robust enrichment at the basal state, remaining there afterwards. So this construct is behaving just like the endogenous protein that I just showed you there. If we then take our ubiquitinated form of that and put it into wild type cells, it's kept out of the cilia at the basal state. It's also kept out after pathway activation. And you might think, well, maybe this ubiquitin destabilizes this protein, and that is true. There is less of this protein in the cell than there is this. But we can find plenty of examples where we can see it in intracellular stores, and it's still kept out of the cilia. But even more importantly, if we take this construct and put it into a 27 mutant, we can see it accumulating in the cilia at both the basal and the induced state. So we further went on and showed that this is also dependent on the BB-some, just like what we had shown previously, suggesting that ubiquitination could really be the key post-translational modification regulating the interaction between the IFT system and signaling. So polyubiquitination or ubiquitination typically happens on intracellular lysines, and there's 21 of those in smoothened. And this is the construct I've been showing you, where we just express this in a wild type cell with no agonist or no sonic hedgehog around. It's kept out of the cilia. If we mutate all of those lysines to arginines, what you can see is at the basal state, it's enriched in the cilia already. So if it can't be ubiquitinated, that's enough to keep it within the cilia. We wouldn't expect all 21 of these lysines to be important, and so we made chimeras and we tracked down that there's just 2 lysines in this intercellular loop 3 that are critically important. And if we mutate the 2 of these to arginines, that's enough to cause smoothen to accumulate in cilia at the basal state. So this led us to the model that we have here, where what's happening at the basal state is that smoothen gets ubiquitinated. That causes it then to be recognized by the BB-some and carried out of the cilia. And that's how smoothen is kept low at the basal state. And then after pathway activation, we would assume that this ubiquitination no longer happens and the smoothen would be allowed to stay in the cilium. So we published that at the beginning of COVID, and that was a point in time which a new postdoc joined the lab and wanted to figure out what this hypothetical E3 is that would be in the cilia that would be regulating this ubiquitination of smoothened at the basal state. So to do this, he created a cell line where he could monitor the state of the hedgehog pathway as well as the ciliary levels of smoothened. To do this, what he did is he took a nuclear targeted GFP and put it under the control of a Gli responsive promoter, and he expressed that in our cells that had the smoothened flag in them. So at the basal state, these nuclei are dark and there's no smoothened in the cilia. After you activate the pathway, the nuclei become bright green and the smoothened accumulates in the cilia. So this reporter, he designed it to be a flow assay. So at the basal state, if you measure the fluorescence intensity by flow cytometry, you can see that the reporter has about 10 to the third peak fluorescence there. After pathway activation, it moves up about 10-fold to a little over 10 to the third. So he had a good, robust reporter to look at. Then he needed candidates to try to figure out what this E3 was. So to do this, we looked at the intersection between ubiquitin-related genes and genes that have something to do with either cilia or hedgehog signaling. This is a list of about 2,000 genes that I've curated from the literature based on large-scale screens of hedgehog signaling or proteomic analysis of cilia. This is a list of about 900 genes that have something to do with ubiquitin. What we found was there were 147 genes in the intersection here. So these are ubiquitin-related genes that have somehow been connected to either cilia or hedgehog signaling. So Bo took these 147, made 3 guide RNAs to each of them, and then put them into his reporter line and measured the fluorescence output at both the basal and the induced state. He then plotted that data on this plot, which looks kind of intimidating, but it's actually not that big of a deal. What he did here on the x-axis is plotted the fluorescence intensity at the basal state relative to the controls, and then the y-axis is the fluorescence intensity relative to controls, but after pathway activation. So if the guide RNA didn't do anything to the pathway, the point should fall right here on the 0-0 intersection. You could see all these gray dots here. So the majority of his guides didn't really do anything to the pathway, but there were a cluster of 2 sets of genes, one up here in the upper right quadrant, another one down here in the lower left, that fell outside of our cutoff there. So this upper right quadrant is typified by patched 1. So this is the 12 transmembrane receptor. It's the negative regulator of the pathway, and its loss increases the basal state, which you can tell by moving in this direction on the pathway, and it increased the induced state, which you can tell by moving up the chart here. So these guys here, when they're knocked out, you increase either the basal expression, the induced expression, or both in that case. These guys down here, typified by things like IFT27, which prevent activation of the pathway. So these guys here are ones that are needed in order to activate the pathway. So these are just hedgehog-related genes that also had some connection to cilia, but we didn't yet know whether they affected smoothened distributions. So Bo went and measured the number of smoothened positive cilia in each of the populations of cells that were knocked down for those genes that fell outside of our cutoff. Again, the majority of them didn't do much to smoothen levels, but 9 genes fell above our cutoff there and had increased levels of smoothen positive. I want to point out that this was done on populations. So you can see our 27s and our patched here are giving us about 30% positive cilia. If these were done in pure populations, this would be about 100% of the cells would be positive there. So these are all ones that fell above that point. So those 9 genes that fell above that point fell into 4 different categories of ubiquitination genes. First of all, we had a group that are scaffolding genes. So these are genes that aren't needed for enzymatic activity, but they bring together the other enzymes and help them to do their functions. We then had 1 E2 ligase, UBE2L3. We got 4 E3 ligases and 1 deubiquitinase. So we took each of these and we tagged it and took a look to see whether or not it was ciliary localized. And it turned out that 3 of these proteins were ciliary localized. I'm going to tell you 2 short stories here in the last couple of minutes that I have. One on UBE2L3 and one on WWP1. So the mammalian genome encodes about 40 proteins that have E2 activities, either for ubiquitin or ubiquitin-related proteins. Bo knocked out all of those, and only UBE2L3 was needed for localization of smoothened to the cilia. So UBE2L3, the mammalian genome or mouse genome, encodes 3 transcripts that encode 2 different proteins. So one protein is 154 amino acids, another 122 amino acids due to the lack of this exon here and a slightly different exon 1. So Bo took each of these 2 and then expressed them in cells. What you can see is that the long isoform is kept out of the cilia, both of the basal and the induced state, but the short isoform is enriched in the cilia under both conditions there. So then Bo took these 2 and used them to rescue our knockout cells, which have been missing all 3 of these transcripts. So our controls shown up here on the top, you see the green cilia, normally under control with no ligand. There wouldn't be any smoothened there. If you knock out all 3 isoforms, you can see this robust enrichment of smoothened in these cilia. If he puts in the long isoform, it doesn't really do anything. We have maintained the enrichment of smoothened inappropriately there. But if he puts in the short isoform, it completely rescues the phenotype there. So the phenotype that's rescued is the one that's ciliary localized. So we think that's good evidence that this ubiquitination needs to occur within the cilia to control the localization of smoothened. So we obtained 4 E3 ligases, WWP1, MGRN1, ARIH2, and MAEA. So while we were putting this paper together, Raj Rohatgi's lab published on MGRN1, showing also that it accumulated smoothened in cilia. He put it downstream of the cilium, probably between the cell surface plasma membrane, non-ciliary plasma membrane, and internal stores. We haven't worked on it. Our data would be consistent with that idea. We published a paper that I'm not going to go into on ARIH2, but showing that this actually works at the endoplasmic reticulum to control the amount of smoothened that's made. When it's missing, the cell makes a tremendous amount of smoothened, and it saturates the retrieval pathway. And so we get smoothened accumulating in cilia by an indirect mechanism. We haven't looked at this at all, so I don't know what it's doing. But let's focus on WWP1, which is a ciliary localized E3. So we got very excited about this because it's really critically important for removing smoothened from cilia. So if you look at our reporter line here, again, the green cilia, no ligand, there's no smoothened in there. You add ligand, you get a huge amount there. IFT27, what started this whole story, you see a large amount there. And if you look at the quantification here, you can see the basal state, there's very little smoothened in cilia, it goes up dramatically. The 27 mutant looks about like the induced state there. And what we noticed in the WWP1 is it looks very similar to that. So this is a really critically important protein for regulating the ciliary levels of smoothened. Importantly, it's also ciliary localized, as I told you already. You can see that here where it's in the cilium. It's also very abundant cell body protein. But very importantly, it's only in the cilia at the basal state. So once we activate the pathway, it's removed from the cilium. And it's removed from this, or excuse me, it's ciliary localization is dependent upon that 12 transmembrane receptor patch, because if we look in patched 1 knockout cells, it's missing from cilia at both the basal and the induced state. So the reason that we did that experiment is WWP1 gets its name because it has a WW domain. WW domains typically interact with short motifs called PY motifs, and there's 2 of these in patched. And I won't go through all the data to support this, but what Bo showed is this WW domain can interact with either of these 2 PY motifs in patched to regulate ciliary levels of smoothened from there. So that leads us to our current model of what we think is how smoothened levels are regulated during hedgehog signaling. So at the basal state, what we think is happening is that there's an E2 ligase, UBE2L3, particularly the short isoform of this enzyme, is localized to the cilia. It works with an E3 ligase, WWP1, that binds to patched 1, the PY motifs there. So any smoothened that's going to diffuse into the cilia at the basal state is going to encounter this. It's going to get ubiquitinated, and that's going to cause it to be cargo for the IFT system and carried out of the cilia. And that's how it's kept low. Then after pathway activation, the patch leaves the cilia, WWP1 leaves the cilia as well. That allows the smoothened that comes in to accumulate there, become activated, and then activate the downstream parts of the pathway. And the UBE2L3 stays in the cilia after pathway activation. We're currently trying to work out the mechanism that regulates patch removal from the cilia. We think it's also a ubiquitination pathway, but what we do know is that it's not through WWP1. So WWP1 mutants remove patch just fine. So there's another E3 that's probably involved in the removal of patched from the cilia. So with that, I just want to acknowledge some of the people that were involved in this. The original work on IFT 25 and 27 were done by previous postdocs in the lab that have now gone on to industry and faculty positions. The ubiquitination of smoothened was done by Parav Desai, shown here. The identification of WWP1 and UBE2L3 was done by Bo Liu with help from a summer student, Jonathan Benoit. The patch data that I didn't talk about yet is being done by Michael Stuck, shown here. With that, I will take questions.
Thank you so much. Just really very clear, wonderful presentation. We have a lot of people who work on Cilia on Zoom, so maybe we can have a little bit open conversation. Let me start with a few questions that other people get their thoughts. What is the difference between rod and cone cilia? Do we know precisely what is the difference between them? What proteins are present, the same and different? Just kind of general overview, what can you say about those?
I didn't catch the 2 types of cilia that you were.
Yeah, rod and cones.
Oh, rod and cones. Yeah, I have no idea. I mean, we do know, for example, the discs aren't completely enclosed in the cones, and of course, there's going to be differences in the cone opsins there. But anything more than that, I don't really know. I would guess that the basic ciliary assembly mechanism is going to be exactly the same in the two. It's just going to be the more specializations that are affected between those.
So if this is the case, that you have the same cilia kind of, right, in rod and cones, that means only the cargo that is delivered to the outer segments will determine the structure.
Yeah, I mean, that would be my guess.
Vadim, are you there? What do you think? And David Williams?
Hi, Vadim Arshavsky here. What I think is really happening there is that something is missing in cones, something which is required for disk enclosure. Because if you really look at the process of disk enclosure, it proceeds for a while, and most likely it's a process which is driven by oligomerization of peripherin and ROM1. And then the last moment of the disk enclosure, that's not just remodeling of the membrane, but it's an act of membrane scission, which, if anything, resembles endosome formation because disk essentially is endocytosed inside the outer segment. And my best guess is that cones simply do not express something very critical for this final scission event. So what we tried to do, working with John Dam, it was a long collaboration. We tried to figure out if dynamins, which are performing that function in many cases, are doing it in rods, but not in cones. And turn out the dynamins have nothing to do with that. So at this point, I think it's an open question. But I think that the point here is not the structure of the cilium, but rather the profile of proteins expressed in rods and cones. Something is missing, and I think that will give an answer. Long comment, sorry.
But then you have, Vadim, then you have pretty well-defined structure of outer segments, right? You know what proteins are there. Many of them the same, some of them different. So do you see an opportunity? And Greg, again, we want you to be part of that conversation.
Not just
Would it be
Vadim.
possible genetically to change rod into cones and cones to rods? Because that ultimately will be the answer, how they are formed.
Can't agree with you more, Chris. I mean, we now have a very good proteome of rod outer segments. I think we're now just moved with a new generation of instruments. It's not published yet, but we'll show it on FASIP first time and hope to publish it this year, at least to bioarchive it this year. We are now up to about kind of over 5,000 proteins, and I think we are not missing anything in rods. Issue here is that we need to move to some very good cone prep, which of course is not possible to do with mice and rats and simple things we used to. Once we're there, I think there will be a tremendous amount of work to be done by the whole community, from that comparison. But if you have a really good idea where to get a really good cone prep for mass spec, which would be relatively pure, I would love to hear.
What is your 5,000 proteins? Is that outer segments or is that?
We actually have the proteome, including both outer inner segment and connecting cilium. I'm sorry, I'm advertising what we're going to show.
No, I'm just curious whether.
What we went down is that we actually have every single IFT protein, every single BBS protein in the proteome, every single proteome of a centrosome, not a single protein missing. So at this point, we're pretty confident that there's essentially no misses.
Right, but can you tell what's there at the disk enclosure step? That's going to be probably not a very abundant proteins right there at the base of the outer segment.
I'm not ready. I'm not ready to analyze.
I'm sure
Nothing obvious. Nothing
I'm sure
obvious.
it's in your data set, but finding it out of the data set, that's the challenge.
Well, we'll make it public very soon. So I guess, but nothing obvious came up so far. But maybe if something is missing in cones, that will be a good clue.
So shall we ask David Williams to comment?
Okay, two comments then. I think the actual structure of getting the opsin up through the cilium still depends upon IFT, right? So you knock down KIF3A anyway, and you don't get cone opsins delivered, just like you don't get rod opsin delivered. In terms of structure of the outer segment, I think it's been overblown a little bit that everything's open in a cone outer segment. That's not correct in some animals. A lot of them do at least partially close off, and some of them even entirely. Some of the discs do even entirely close off. So, but that's a different question from ciliary transport. That's my
All
two
right,
cents
we have a
worth.
question from Zoom. Mark Krebs.
Yes, hi. Thank you for a wonderful talk. I just had a question about whether or not there's any evidence in the mammalian photoreceptor system for operation of this ubiquitin machinery or elements of it in the outer segments.
Yeah, there is some evidence. The Naturi lab had published in BBS mutants that there was accumulation of ubiquitin in the outer segments. So I think it's probably happening there as well. I mean, Vadim and I have argued over the years about whether you really need to bring stuff back from the outer segment or you just let it go and be shed with the disc. So, but it does appear that in the BBS mutants, you do get a ubiquitin accumulation, the same as you would in a primary cilium if you have a BBS mutant. So I think it's completely plausible that this is also working there in the outer segment to remove things that shouldn't be out there.
Thank you.
Greg, let me ask you one more question. So at that point, you have many of those proteins involved in cilium, connecting cilium there. Do you know their function to some degree? You know, small g protein, calcium binding protein, receptors, and so on. Even if you do not, if you have a lot of unanswered questions yet, can you put mechanistically how that cilium works? In your mind, you know, as a working hypothesis, let's say.
I mean, I sort of can get it at the level of hedgehog signaling, which we don't need a whole lot of structure there. We can put receptors out there and pick up signals and move back and forth from the cilium. It's a little unclear to me why we need to have this dynamic movements. I mean, that's a question that comes to mind. I think we probably appreciate that the cilium is important because you have to reduce cytoplasm so you can amplify signals there. So in that regard, but beyond that, what the cilium is doing functionally, I guess I'm a little unclear there, other than providing a structure to organize sensory transduction pathways. And then, of course, motility, it makes a lot of sense. You have this very organized cilium that generates sliding microtubules to produce spores. So that makes a lot of sense.
Oh, this is really fascinating work. I wanted to ask you, since a lot of the ciliopathy, it seems to be developmental case, what would be the effective treatment strategy for all this, different diseases affected by, caused by ciliopathy genes?
Yeah, I think it's a fantastic question. I'm not sure that a lot of these developmental diseases are not going to be treatable, I don't think. I think the best hope we can do is provide some information on the genes that are involved, and families can make decisions based on that. A lot of them have to do with hedgehog signaling, so potentially you could do something there. In the mouse, it's really hard to come up with developmental phenotypes that you couldn't trace back to hedgehog defects. The concern with if you do anything with hedgehog signaling, you're going to end up with all sorts of birth defects on their own right. So unless you get it exactly right in the right cell type, so I think that's a really difficult problem. I better hope for things like retinal degeneration or polycystic kidney disease. And polycystic kidney disease is one I think is actually really has a lot of potential for treatment, because about half of the patients will live their life and die from something else before they reach end-stage renal disease. So that says to me that we don't need to cure this disease. We just need to slow it down a little bit, and it would have a huge quality of life improvement for these patients from there. But these developmental defects, when you've got a massively malformed heart, I just don't see a lot of treatments that can be done, and especially in a broad way.
Hi, Dr. Pazour. This is Philip Kaiser. First, this was a phenomenal talk, and it's great investigative work to figure out why this is needed in proteasomes, not needed in proteasomes, and used in deuterosomes. My question is about the IFT 2527. So it kind of follows on Chris's comments. So you have a calcium binding protein and a GTPase, and do you know mechanistically how those activities are linked to your observations with with smooth and then related to that, these proteins, I think you said they're present in chlamydomonas, where I don't think these smoothen and is going to be relevant at all. So do you know what role they're playing in chlamydomonas?
Yeah, fantastic questions. The GTPase, we made a lot of mutations in that and never really got a good answer for what that was doing. Probably regulating its formation into the particle itself, regulating at that point, and didn't necessarily seem to be connecting to cargo the way we had hoped it would. The calcium binding, we made a lot of mutations with advice from Esben Lorentzen, who did the crystal structure on that. That was really difficult because the calcium binding is kind of an unusual calcium binding that involved the backbone. And so I'm not sure that we actually disrupted it with our mutations. That didn't, if we disrupted it, we probably had bigger problems with the protein there. So we never really made any progress with that. The question of chlamydomonas is a fascinating one and something I've thought a lot about. So my best guess, and this is purely a guess, is that there was some sort of ancestral pathway that involved movements of signals in and out of cilia. And hedgehog co-opted that pathway when hedgehog evolved. And in chlamydomonas, there is a well-studied pathway. I mentioned it at the beginning, which is mating, where the two mating types come together, the cilia touch each other, and the signals propagated down to the cell body. And that signal movement depends on IFT. So if you stop IFT and you fuse the cilia, the cells don't lose their cell wall and fuse. The problem with that theory is nobody has tested to see whether 25 and 27 mutants affect mating. And the BBS mutants, which I would put downstream of that, I made BBS mutants in the early 90s, so I didn't know they were BBS mutants, and they mated just fine. So I'm not sure that's the pathway, but my guess is that there is some sort of ancestral pathway that involves signals moving in and out of cilia that 25 and 27 are doing. The interesting thing in invertebrates like Drosophila and C. elegans is they got rid of most of their cilia. So most of the cells in invertebrates are not ciliated. Whereas in vertebrates, we've got cilia everywhere. So cilia are present pretty much in every cell type at some point in development and remain in adult organisms. And that's just not true in flies and worms.
Hello, my name is Hewitt. I'm a postdoc in the LabNT lab. I wanted to ask you about phosphatidylinositols. I know that they're important for ciliar formation, but are they connected mechanistically to IFTs?
So I don't know of any data that's connected them to IFT per se. Definitely the formation. They're thought to be involved in regulating cargo unloading from IFT after you get out in that, do you get through the transition zone? So perhaps at that point, but I don't know a lot of work in that space.
All right, Greg, I think we will just conclude with one more question because I feel not satisfied. So mechanistically, this connecting cilium play a critical role transporting all of those proteins. But if you look on rod or cone cells as they develop, they initially have either minimum this cilia or just not at all, and they start growing at a certain point and then expand into rod outer segments and cone outer segments. So the question I have, and again, I'm not that familiar with this area of research, do we know the sequence of expression of those genes? They are ciliary genes.
I don't think we know it precisely in photoreceptor cells, but there's a paper out of Peter Jackson's lab. I think it's in bioarchives now. Actually, it might be published now out of bioarchives, but they very carefully looked at the proteins that appeared during ciliogenesis. And what they found, which I found really remarkable, was IFTB came first and then IFTA came later, which I had prior to that work, I thought these would have come together, but B shows up first at the base of the cilium, and then A comes later, and other proteins come in to the cilium later on in the sequence of events. But
so if you will compare this expression with the structure, that could provide some information about what role they play in the cilia formation, right?
Yeah, although I don't know that you'd have the temporal control, the temporal precision to do that, because cilia assembly is pretty fast. So once they decide they're going to build a cilium, it's probably an hour or so before there's a connecting cilium done. Are you going to be able to get the RNA expression with the precision for that? I'm not sure that you would. I think probably proteomics and immunofluorescence would be a better approach to figure out the sequence of how it's coming in.
All right. Greg, thank you so much. Thank you for being with us, and great presentation, very important area of research. So we are extremely grateful. Please join me to thank our speaker.
Thank you, guys. I appreciate it. This is great. Lots of good questions.